Hi there. So this video is about this one here, D2, which is to collect sufficient valid data for all samples with appropriate precision. Okay, so this is quite tricky. It's not really like you're writing a sentence as such, but it's just considering that you've done enough experiments really and that your data is good enough. So one thing you could do if you had time is you could repeat the experiment. So you could do something 3 times and that just increases the reliability. If you don't have time, you can collect a classmate's data, a peer's data, and compare it to yours, and that checks something called reproducibility. So, for example, for the reagent test strips, you could add a column for your peers' results or a few peers' results, just to double check that what you got was right. It makes it more reliable if they got the same as you. So you could add that in if you wanted. You could also add a paragraph to explain that you understand it would make your data more reliable if you had repeated, but however, there's a cost to it. For example, you could have repeated the agar plates, but the cost to the college of everyone redoing their agar plates costs a lot of money. And so that, and in terms of the time, especially as that one needs to incubate, might mean that you didn't, okay? There's a table here which is just a few ideas of what makes, really good data. So, for example, did you do the tests on all the samples? So, for example, did you do your reagent test strip, your Virec Benedicts and quantitative Benedicts? Did you do it on both urine samples? And did you do, obviously, your microbiology on the microbial cell sample? Then that's probably sufficient, because you haven't got anything missing. Okay. In terms of validity, good validity, it basically means that your procedures were right and that you controlled the variables well. So if you controlled the variables other than the thing you're investigating, then your results should be valid. If you got lots of contamination, then your microbiology results might not be very valid, in which case it's worth definitely having evidence that you have looked at somebody else's results so that you've got some valid results to make the conclusion on. Okay. In terms of precision, it's that your measurements were made really well. So, for example, did you read the colours accurately from the lookup table? If you had any repeats, was each repeat similar? You could talk about in your quantitative biology, when you shared class data, were the standard deviations small? Did you use the right units in your results tables? And in terms of recording, your results tables should all be there, all filled in, all correctly filled in. Okay, so it is a really tricky one to kind of sign off, if you like, and it is a distinction standard. But generally, if you did what we asked you to do, what we suggested you did, and you did all your experiments, you've got all your results table and you filled it all in correctly, and you've compared to other people's results where maybe you think yours weren't top quality results, then you should pass that D2. If you really wanted to kind of cover your back and write a paragraph about why you think your results are sufficient, valid, precise, and well recorded, you could write a paragraph to explain that. That's up to you. Or otherwise, we will just look across your whole assignment and decide if we think that you collected sufficient valid data with appropriate precision. Okay.